Good afternoon, everybody. It's my pleasure today to introduce Dr. Ladislav Hamerlík from University of Banská Bystrica and National Academy of Sciences of Slovakia. Laci is a paleoecologist and aquatic ecologist, mostly interested in reconstructing climate and environment change in the past using biotic proxies from the lake sediments. He did his PhD in University of Zvolin in Slovakia, and after that he did postdocs in universities of Copenhagen, Lund, and National Academy of Sciences on Poland. And today Laci will introduce us to the using a paleoenvironmental archives of the lake sediments to learning about past and the future of our environment. Thanks, Laci. The floor is yours.
Thank you very much. So, before I start, I would like to tell you that I brought some sweets, so when you are leaving, feel free to get. I'm not sure it will be enough for everyone, so be fast, but don't do it now, okay, because it would be kind of disturbing. Anyways, thank you very much for the opportunity. I'm really happy that I can be here. I love Sevilla. It's my third trip here, and I'm super grateful to Victor because it was Victor's idea about this size-temperature relationship, and we should test it, and then we need those training sets and all that stuff around. But obviously I love to come here, and now I even can have a presentation, which is very nice because I like to talk about my research focus, which is paleolimnology. Even though I started as an aquatic ecologist, it's still aquatic ecology in a way, focusing on chironomids. So I will talk partly about paleolimnology, why is it so interesting, how we can use lake sediments to reconstruct past environmental changes, a little bit about chironomids, which is my main proxy I'm using and my team is using. And I will show you I will demonstrate paleoecological reconstruction on our own data. So, before I start, I would like to show you some pictures. Where am I coming from? That's the University of Banská Bystrica in Slovakia. This is not the university, obviously, but it's a very nice building. So I wanted to, I mean, you don't want to see the university building, so actually I always put this one because it's beautiful. Victor has been there. It's a very nice...
It's a tavern.
It's a... Victor... Yes, exactly. You can have actually a very nice little town, about 60,000 people, and it has a big university, so it is quite lively, and it has a beautiful position at the foothill of 3 mountain ranges in the center of Slovakia, and I like it as well. I mean, it's a different story here in Sevilla. It's a huge city to live in. At the same time, it's very close to the Tatra Mountains, which you may know. This is the highest part of the whole Carpathian mountain range. There are very high mountains also in the Romanian part, but this is actually per se really the highest peaks of the whole Carpathians. And if you're a paleo thing and it's lakes, you need a lake to reconstruct lake sediments, right? So it is very convenient to be in Banská Bystrica. It's a 2-hour drive to the Tatra Mountains, which is actually full of lakes because it was glaciated. So there are like 90% of all lakes in Slovakia, all natural lakes of Slovakia are in the Tatra Mountains. So if you are a paleolimnologist, it's a great locality to be. So when we want to understand how lake sediments are working, how they are formed, and why are they so good for reconstructing past environments, we should understand what is a lake. And if we look at a lake for thousands of years back, it's a beautiful lake. We don't really know that much about the lakes. People didn't really know much about lakes. It was a huge body of water with some strange sounds, changes in the color and the strange things happening on the lake. But we didn't really know what is happening, what is going on in the lake itself. So, of course, every noteworthy lake has its own monster. If you don't have a monster, it's not a real lake. I did some research, and really, I mean, I don't know how it's in Spain. I hope that Doñana and other places have their own monsters. If not, I'm sorry for you. But you should have. And I don't know if you know, but this guy was even described in nature as a real species. This is the Loch Ness Monster. Or if you live in Central Europe, where there is a huge tradition of fish ponds, its own Wasserman, which is a creature, waterman, who is living in and taking care of the pond. Really, literally every single one. No one saw them. I mean, people are disappearing time to time. So in public Germany, Hungary, you will find those guys living in lakes. And if you go to Asia, South America, you will find gods and goddesses taking care and living in the lake. And it was funny, in a very fortunate period of my life, I was part of a Danish team with a lot of money. So we went to Tibet to study glacier streams and lakes. And we really wanted to take some samples from this lake. We came there, and it turned out that it's a holy lake. And you are not supposed to take a single life from a holy lake. So we were not allowed to sample it. We didn't even try, because people let us know that it's really not appropriate. And I mean, they wouldn't kill us, but I mean, you don't want to mess up with locals. And they were very sweet people. We got even yak, the black tea with yak milk and salt, which is a local thing to get for visitors. Anyways, we didn't sample this lake. We sampled many other lakes, not this one. And if we look at a lake, we can look at it from a very philosophical point of view as well. And I really, really like this part of the lakes, because it shows that lakes can inspire artists, painters. The whole school of painters in Denmark are painting just lakes and marine environment and seas. And I really like this. You most likely know Walden, this book from Henry David Thoreau, who was a famous American philosopher, eco-philosopher, let's say, naturalist, and he perceived a lake as a mirror or an eye of the earth through which we can see our own depth, which I found beautiful. It doesn't say a lot about the lake from scientific view, but it is nice that lakes can inspire people. And if we want to look at a lake as a natural entity from scientific point of view, we have to go back to Forel, Alphonse François Forel, who is the father of limnology. And limnology, of course, is the research of lakes and the science of lakes and continental waters in general. So, what is a lake? A lake is a, let's say, body of water surrounded by land. It's true, but a puddle is also a body of water surrounded by land. So it's a little bit more into it, right? So a lake always occupies a basin, and it cannot be separated from the catchment, meaning that we cannot really understand a lake, what is happening in a lake, know the catchment. And the catchment is geology, vegetation, age, climate, local things, global things. All those things together are creating the catchment. And it also always has a structure of water mass. We know that there is a littoral zone, a profunda, a sublittoral, there is a hypolimnion, epolimnion. So there is always a structure which we can study, and which Forel studied, even though he was a doctor, but he decided to dedicate his life to limnology, which is amazing. And every lake is a product of age and origin of basin. And climate, that's what I said, that it's actually, it has to be in a context of the whole catchment and its age. And we can study every lake from paleolimnological point of view, meaning that we can look at the lake and reconstruct the whole history of the lake. And the history of the lake will mean also the history of the catchment and global changes. For that, we need a lake sediment. Yeah? So, is looking at a lake as a huge sedimentation trap, meaning that everything what gets, or almost everything what gets into the lake will sediment, will accumulate at the bottom. So we have a lake which was just formed, and suddenly it is somehow occupied by animals, by organisms, by species, different species, but also it will, so if we look at a lake, and we will always be able to see all the changes, all the things that are in the catchment. So if there is a volcano in the catchment, it doesn't have to be in a catchment, but close by, we will always see tephra layers, which if the volcano after the eruption hits the lake surface, it will accumulate, it will stay, and it will create simply a layer in the sediment. If there is a glacier in the catchment, it will always bring glacial sediments, which is different from the normal sediment from the catchment, and it will accumulate very specific glaciated laminated sediment. According to that, we can see that there was a strong glacial influence. It's not always, it has to reach some strength of the influence, but if there is a strong glacial influence, you will see this laminated sediment, which is really amazing, and I can show you one from the Tatra Mountains. Of course, if there is human influence polluting the lake, you will see it in the lake sediment. Plus all the vegetation, because the pollen is hitting the lake surface, and then it accumulates in the bottom, creating layers after layers, all those things all together. Plus we have a community, a lake community, and the lake community is always somehow reflecting the environmental or the nature of the lake. If we have a very cold oligotrophic lake, the species will be also those that are somehow living in cold oligotrophic environment. If the temperature is changing, all the trophy is changing, or pH is changing, also the community is changing. So you will have different species in a warm lake than in the cold lake, right? So all those things, all those zooplankton, phytoplankton, and benthic organisms will die at some point, and they will sink on the bottom and again create a layer of sediment. All those things together from the catchment, from the biota of the lake, they are accumulating on the bottom and creating layer by layer a yearly archive with a very high sedimentation rate. So it is well, that's not what I want to say, but doesn't matter. It is creating a continuous archive. That's the lake sediment. It's really, really cool archive to study environmental changes in the past, right? So what we have to do to study that archive, we have to take a sedimentation core. This is the core. This is what I was talking about. This is from that part of the lake when it was under a very strong influence of a glacial, and then appears, and then this is non-laminated gytja, so organic sediment, organic rich sediment, lacustrine. And then we can cut it into layers. It depends. If it's a long core, we usually cut it into 1 centimeter layers. If it's a short core, we should usually cut it into 0.5 centimeter layers. And then of course it's a higher resolution if it's a 0.5 centimeter layers. Then we should somehow analyze all the proxies ideally all the proxies that we find in a lake sediment. And in a lake sediment, there are so many different proxies, especially now when we start using DNA methods and DNA analysis. It is much, much more than what we see as micro and microfossils. But even using standard old-fashioned methods, we can see remains of so many different organisms, like aquatic insect remains and foraminifera. This is from a marine sediment, of course, and mollusks and cladocerans and the diatoms and oribatid mites and so on and so on. So there are plenty of different remains, organic remains, plus there are, of course, physical remains and chemical remains that we can analyze, and they can give us a clue to reconstruct what was happening in the lake at that point. And then, based on those data, we can reconstruct the community changes over the period which we are analyzing, which can be hundreds of years, it can be thousands of years. It always depends on the age of the lake and the study, what we want to study. If we want to study human influence, it's usually hundreds of years. If we want to study Holocene climate change, it is thousands of years. And we can do it both qualitatively, so simply describing what was happening, and we can do it also quantitatively through quantitative reconstruction. And when we do quantitative reconstruction, we are actually inferring the temperature or pH or other environmental parameters. Yeah. So the first step of every paleolimnological survey, of course, is finding a study lake. And you have great limitations here because for that you have to have a lake. If you don't have a lake, you cannot do paleoecology or paleolimnology. You can do paleoecology, you cannot do paleolimnology, but it doesn't have to be actually a recent lake. It can be also paleo lake. It's important that you get a lake sediment, which is not existing anymore. But just to show, these are the localities of the Slovakian paleolimnological research for the past 25 years, let's say, which is actually the whole time of the paleological survey in Slovakia. And as you can see, 90% of all localities are concentrated in that small spot, which is the High Tatra Mountains. So we have lakes here, we have some artificial lakes here, which we studied for human influence. Then we have a Danube with plenty of oxbow lakes with lake sediment, kind of, which we studied. But most of the paleoecological survey in Slovakia is concentrated to the Tatra Mountains because we have lakes there. Otherwise, if we don't have lakes, we cannot do it. At the same time, those lakes are remote lakes, high mountain lakes of glacial origin, so they are about 13,000 years old. So they are very, very good for quantitative temperature reconstruction. That's another thing you have to consider. What do you want to study? If you want to reconstruct pH changes, you have to find a lake from about, that actually it went through some pH changes. If you want to study climate change, you shouldn't study a lowland lake with a very strong human influence. It should be a lake which didn't get too much human influence, ideally non-human influence, and you can see the temperature signal very strongly. Then you have to take a sample, and that sample, the sediment core, can be a long core or a short core. Again, it depends. If you want to study whole Holocene changes, you have to take a long core, which takes much more money, much more time, much more equipment, and so on. If you want to study a couple of 100 years back, it's enough. It's much easier. You get this kayak sediment corer, which is a gravity corer, and you can do it even by hand if you have a very, very shallow lake or a pond. However, for example, this short core, it's 60 centimeters long. It comes from Greenland, and it represents the last 2,000 years of the lake. So you can see the sedimentation ratio is very, very low sedimentation rate. And then we cut it. It's one of the best studied lakes in Slovakia. I will show you some studies from there. But these are short cores which cover about the last 700 years. It's also a mountain lakes from a mountain lake, so the sedimentation rate is also not that high. However, we have lakes, for example, from the Banská Štiavnica region, from man-made lakes. Their 200 years is a 1.5 meter long sediment. So you can see that it's the sedimentation range or the sedimentation ratio is changing so much among lakes. Yeah, so you have to consider that as well when you are deciding what method you should use and what you want to see or what you want to reconstruct. And we are cutting it in usually 0.5 centimeter layers on site, unless we decide for long cores. If you take long cores, actually there is almost no limit how much sediment you can extract, because with this method you are taking 2 meters from a floating platform. It's a piston corer. You can take 2 meter pieces one by one, and you can go as long as the sediment allows you, which can be easily 16 meters. We extracted 12 meter sediment core from one lake in Slovakia. From Greenland it was, from Iceland it was 16 meters. So actually the limitation is usually the bottom of the lake and the beginning of the lake formation. This is from Greenland. It's a little bit different method. You can do it from ice as well, which is much cheaper. This floating platform is about 700 kilos. If you have to carry it to a lake in the Tatras where you have to walk for several hours, it will take you 2 days to just bring the disassembled floating platform piece by piece, even using Sherpas, because some of the pieces are super heavy. So just taking one sample, one sediment sequence can take 1 week easily. And then we bring back the sediment sample, the sedimentation core, and we cut it usually in 1 centimeter piece samples. This is again from Popradské pleso. This is the part of the sediment which was under very strong influence of glaciation over a glacier in the catchment. And this is the last, I think, 8,000 years. This altogether, it's a 6 meter long sediment sequence. And then the next step is analyzing the proxies. So you have to somehow extract the proxies. If you are studying chironomids, you have to pick up head capsules. If you are doing cladoceran remains or diatoms or pollen, it's much easier. You just mix the sediment and put a drop of it, and then you can start identifying under the microscope. I want to demonstrate this process on chironomids because I'm working mainly with chironomids, even though in the lab we are working also with the cladocerans and diatoms and pollen. But this is my favorite, I'm sorry. And talk about chironomids because people usually tend to think that they don't know chironomids, that they never met them, they never seen them, but actually everyone knows chironomids, because if you have a fish tank, you are feeding them with Chironomus, I don't know, it's Stumi, Plumosus, whatever, a bloodworm. If you are catching fishes, you might use them as a bait, and if sometimes this is from Lake Victoria, but once they are swarming, they can create those huge groups, swarming groups, which look like actually smoke on the water. And however, they are of course insects. They are very tiny. They are everywhere. There is almost no water body which wouldn't be colonized by chironomids, literally. They can be quite extreme, but that just talking about extreme species of chironomids would be enough for a separate talk. So I'm not going to go into that, but they are holometabolic insects. This is the imago. They have 4 life stages: the egg, the larvae, the pupae, and again the imago, of course. And this is how the larvae and the pupae look like. But this is interesting. And those, of course, are aquatic, in aquatic phases of the life cycle. Usually aquatic. There are some terrestrial species, but the most of them are living in aquatic environment. And this is what we are interested in when we are doing aquatic ecology. But when you are a paleoecologist, you will only see this part of the head capsule, which is heavily sclerotized. And it remains in the lake sediment forever, almost. So why are chironomids good proxies, and actually what is making a good proxy a good proxy? So there are several, of course, points you should meet as a good proxy or good bioindicator, let's say, because it's even a better word. It's a high abundance, and it is high taxonomic richness, very different ecological requirements, short life cycle, so the group can react or respond to changes of environment, and then good identification. So all these are, I mean, chironomids are meeting all those points, but there are many other groups that are meeting those points. However, they don't leave any remains in the lake sediment, like Oligochaeta, a huge group of great indicators, plenty of species, really thousands of species, but they disassemble totally. And unless you are using molecular methods, which is still not standard, you will not find the clues of Oligochaeta, even though they could be good indicators. For being a good proxy, paleoproxy, you also have to somehow leave some sign, some mark in the lake sediment, like chironomids. So they have sclerotized head capsules, and then even the larvae is disassembled, the head capsules stay in the sediment, and they can be identified very nicely. And also, you don't have to, but the group should be numerous in the sediment. I showed you that we are taking those cores, and then we are cutting them into 0.5 centimeter or 1 centimeter slices. So we want to reconstruct that slice, and based on that slice, one slice, we should reconstruct all of them, but from one slide we want to do chironomids, diatoms, dating, loss on ignition, blah blah blah. So you can imagine how many remains should be there. So if we take it apart, that sediment, it's really just a tiny little sediment we get for analysis. In that tiny little sediment, we should find at least 50 head capsules. Yeah, at least. Ideally 100, let's say, or even more, but 50 is the least to do a reliable reconstruction. And chironomids these are recent head capsules, which I took in Greenland, and I had to cut the head off. So these are recent head capsules from Greenland, and these guys are 13,000 years old head capsules. So you can see there are plenty of tiny structures, kind of important for identification, but for genera or morphotype identification, you actually require the mentum and the mandibles and, let's say, occipital margin. So if you have those three things, which are almost always part of the subfossils, you will be able to identify those remains. So chironomids are really, really cool. And you can see that there is no such a big change in the consistence of the head capsules even after 13,000 years. And this particular species, for example, it's Heterotricycladius grimshawi. This lives now in a very cold lakes of Greenland, Norway, Iceland. This was found in the Berling Oscillation in Denmark. So this species is not there anymore. So it's also showing that they are really reflecting the local conditions. Yeah. And if you want to do a thorough, complex reconstruction, of course, you shouldn't rely on one proxy, because every type of proxy is influenced by different environmental variables. If you are working with chironomids, you are working with animals that are living on the bottom. But it's interesting to include also planktonic organisms, right? Because they are more controlled by, for example, light availability and nutrients. Or if you want to have a vegetation signal, what was going on in the catchment vegetation, you should also include pollen and or cladocera if you want to see what was happening with the fish and water currents and transparency. Then you should also include, for example, zooplankton, cladocera remains. It also means that your team should be much bigger, and it takes much more effort to do the reconstruction, much more knowledge, it's much more complex. I'm sorry. But, so, let's say you want to reconstruct, because that's the next step, of course. You have all the analyses, you did all the statistics, and now you actually want to say something. What was going on in the reconstruction? You want to reconstruct what was going on in the lake over the last several thousand years, for example. And then this is how it looked like in reality. But what you get is this. So you get actually all the pieces of it, but they are not really showing the real picture. You get something, you get all the, actually you never get all the species, because there is such a thing like taphonomy. So part of the animals will be destroyed, the part will be taken with the outlet stream. Some will be eaten by scavengers. Some will be petrified, and so on and so on. So you will get something, but you should really think of a complexity of the changes. So what you see in the proxy data, it's not all the species they were living there. Plus, you will have species from outside of the lake, meaning from the inlets, from the littoral, which is part of the lake, but it's not the profundal. Anyways, it's very complex. It's also very beautiful, but it can be quite challenging, because if you do the reconstruction, and for that, because you have to simply put the puzzle together, and for that you should know a lot about the ecology of the species. If you know an ecological requirement of the species or the morphotypes, you will be able to do something like that. It will be not perfect. It might be, you might not see the details, but you can reconstruct the original environment. But if you don't know, or you misidentify something, you can put something together which will actually make sense. It will, oh, wow, it looks like we can reconstruct something. It's just not how it looked like. And you will use all the knowledge and all the tiny little pieces, but it's easy to make a mistake sometimes. And now I will demonstrate some papers, obviously, what we did in the Tatra Mountains, not only the Tatra Mountains, but for example, this lake in the Tatras, it's called Popradské pleso. And we are analyzing it with Victor as well, right, for head capsule size and temperature changes. But we also did an analysis, several analyses. It's a very good lake for paleoecological reconstructions, because it's one of the oldest lakes in the Tatra Mountains. It's about 1,500 meters. It's a deep lake, it's a big lake. And for a long time, it had no human influence at all. The last 200 years, people started to come there, and they started to build hotels by the lake, and very cleverly started to pollute the lake directly. I mean, they put a pipeline into the lake, which is, I mean, very nice. At the same time, after several years, they removed it, but still, we know that all those things were happening. So the lake is great for studying long-term climate change over the Holocene, because it's a very old lake. It's great to study glacial influence, because it has had a very strong glacial influence for a long time. But it is also nice to study human influence, how we can screw a lake's ecosystem by, especially if such a oligotrophic, very cold system, by putting extra nutrients in it. So one of the studies was, so we have several cores. This one is for we analyze one long core and several short cores. And from the short core, we try to analyze the Little Ice Age. But what is more interesting, this upper part, which is about 10 centimeters only, we were curious what was happening with the biota of the lakes when people started to pollute it. And then we did this multi-proxy study, and it's showing, the results I think show how important it is really to do multi-proxy instead of just looking at one proxy, because it is really giving us much more complex picture. So as you can see from this, these are PCA first axis scores, so it is showing the whole community change, right? So as you can see, when there was no tourism, and there are Cladocerans, diatoms, and Chironomids, and this is lost on ignition. So no change, no human influence. Then people started to visit the lake, and they started to build even some chalets and houses, wooden houses, but not polluting the lake directly. At some point, however, they built this nice big hotel, the wastewater into the lake, which was very clever, of course, and then the change is starting. And it's very interesting because Cladocera and diatoms, they started to react right away at the same time, which is interesting. But Chironomids, they didn't even notice it, or just some of them somehow, like, I mean, not a big deal. So for Cladocera and diatoms, they are controlled directly with the nutrients, while Chironomids are much more into hypoxia on the bottom. They didn't really notice the change until it started to cause hypoxic conditions on the bottom. And that's the nice thing. That shows that it's really nice and very important to do this complex reconstruction. Also, what is interesting, that after the pipeline was removed, Cladocera and diatom communities didn't change. They didn't go back to the original. However, Chironomids started to move back to the original stage. They didn't reach it. It's still, but they reacted upon it, which is interesting. So our interpretation was that Chironomids responded to oxygen depletion caused by pollution, but not pollution per se. Another study from the same, but it's a long core, was the study of what was happening during the deglaciation. So this is the glaciated part of the sediment. It goes much further down. I just don't show it because it's not that important, because the community is always made of two dominating species, Pseucrocladius nivosa and Micropsecra radialis type. It's not that important, but it's a very low diversity and a very typical, extremely cold stenothermal community. And then when the glacial disappeared, it shrinked and then disappeared from the catchment, the community suddenly changed. So this is the community of the glacially impacted lake, and this is the community of the kind of a modern community which remains until now. However, so we described how the communities were going further and further, how they were actually following the glacial. There is no glacial now in Slovakia, in the Tatra Mountains. but we still have lakes with that community with very low diversity and very dominated by these two species. Interestingly, they are not necessarily in the uppermost lakes, but they are always in the coldest lakes. And the coldest lakes don't have to be actually at a very high altitude, but they have to be shaded. And we did temperature studies and shading, local shading is such a powerful factor cooling down the lake. We have two lakes next to each other. One is shaded, another one is now not. I don't have the picture right now, but in end of July we took a picture. One is completely frozen, completely. The second one is without any ice. So this gives us hope that those communities that are super vulnerable, and they will be injured, of course, and warming, they can still survive in not the uppermost lakes, but in the most shaded lakes. So this is also an interesting result. And then the lakes I mentioned previously, we have this very interesting city which is called site. It's a very beautiful city town, let's say, in Slovakia. It's pretty small, and what makes it interesting that it's an old mining city built by Germans about well, it's not 300 years ago, but what was built 300 years ago is the system of lakes, artificial man-made lakes, and they used those lakes as a power supply or a source of energy source for the whole mining machinery, because gold and silver was extracted there. 50 lakes which they built, still about 20-25 lakes are remaining in the region. They are really beautiful. They are used for very different purposes, from angling and recreation to like drinking water supply. But we were interested what was happening during the time when, after they built, or after the lakes were not formed, they were built actually. What was happening with them, and we did this multi-proxy study using chironomids, the diatoms, and cladocerans plus pollen to describe complete change or complex changes in the catchment and in the lake itself. And we distinguished three phases. In the first part, the lakes were used as energy source. It means they were using the water, the bulk of the water, almost on everyday basis. So they will let it go. They will, well, probably not everyday basis, because it was not that easy to get the water back to the lakes. But they were super dynamic. So they were dry for a lot of time. They were using as power source. So they were not stable ecosystems. And then we can see it also on the lake sediment that there are very few remains. It is partly because there was not that much life in it, but also because it was drying out. So even if there was something remaining, there was, because I mean lakes are always full of life, but this life takes time to build, right? So in this first part, we see energy source. There is water level oscillations, very severe, no stable community, and the catchment was barren. They used all the wood in the catchment to build the construction in the shafts, and also to simply burn it. And then when they stopped using modern energy sources, it was a stabilized hydrology. The community started to succeed, and meadows appeared. And the last phase is the recreation, when there is no more mining in the region anymore. So we can see very strong nutrient enrichment, reforestation, and and people started to build houses around, so there is a very high organic pollution in those lakes that they are not used as a water source, of course, drinking water source. And other, I don't know what is, okay. So, you can use chironomids also to reconstruct fish introduction, and the interesting thing is that the same signal can be very different for different lakes. So in this particular lake from the Tatra Mountains, it's an oligotrophic, very cold mountain lake. And there is this group of chironomids called Tanipodine, and they are free-living, big larvae, so very attractive for visual predators, for salmonid fish, for example. And in this case, we interpreted these places where Tanipodine are entirely missing, that it is the place where the fish population was very strong, and they started to do the introduction of the fish. This is originally a fishless lake, I have to say, which I didn't tell you, but it is because most of the Tatra lakes actually are fishless by nature. There are only 2 lakes that were introduced by fish naturally. So here, Tanipodine absence is connected to presence of fish or high population of fish. Another lake, though, in other study where what we did in the Lake Balaton, you might know it, it's the biggest lake of Central Eastern Europe, let's say. The Tanipodine presence was actually the indication of the presence of fish together with very low chironomid concentration and taxonomic richness. And why is that? Because in the previous lake it was the opposite, right? Here it is because of the system of the lake. This lake is not a clear water lake. It's not a beautiful oligotrophic lake with great visual properties. This lake is actually a very turbid, shallow lake. So the visibility is very short, very low visibility. So here there are no visual predators. The fish population living here actually is carp and bream, common bream, and those are just scavenging everything what they find. They are benthic feeders, so they are feeding on benthos and not visually finding the prey. So these larvae are actually free-living, and they can avoid being caught by the fish this way. So this, just for the demonstration, I wanted to show you that the same effect, the presence of predators or presence of one group of chironomids can be interpreted totally different way in a lake which has clear water and in a lake which has a totally different system with low visibility, high turbidity. And okay, that was it. I mentioned that you can reconstruct path changes also through transfer functions as quantitative reconstruction, and it works. For that, you have to build a training set, and a training set is basically a group of lakes. The bigger the better, at least 50, let's say 70, 100 would be great, where you have a strong gradient in one environmental factor. You should find, for example, it's usually in the mountains, it is also a pH gradient or productivity gradient, yes, so phosphorus amount gradient. So you can, during the transfer function, the taxa from surface sediment samples are related through statistical methods to environmental parameters. And then, using that knowledge, you can quantitatively reconstruct the that particular environmental parameter. As I said, it doesn't have to be temperature. Usually it's temperature. The most of the environmental reconstruction, quantitative reconstruction in paleoecology are focusing on temperature, but it doesn't have to be temperature. It can be any other thing which is driving the community in your training set. So actually you should build your training set before you want to do the reconstruction, and you should know which is the parameter you want to reconstruct. Because having a strong temperature signal in your training set will not allow you to do a quantitative pH reconstruction, for example, because maybe it's not even there or it's not represented. However, the fun thing is that if you do temperature reconstruction, you also have to be very careful with the lake. You have to choose a lake which was not influenced by other factors or just minimally, but the factor of change, a driving factor of the community was temperature or climate change. Because if you have any other things, any other local drivers, for example, that are not connected to temperature, it will give you a temperature signal anyways, and you can reconstruct it in a wrong way or interpret it in the wrong way. Meaning that just because in your quantitative reconstruction you will have a higher or lower temperature, it doesn't mean that the temperature was really changing. It could be that some other factor was changing, it changed your community, and still you will perceive it or you will see it as a temperature signal. Just one example, we did this study on Lake Lögorin, which is one of the big lakes of Iceland, which is influenced by a strong glacier from Vatnajökull glacier, from the Vatnajökull ice cap, and this is a surging glacier. And what is a surging glacier? So some glaciers have the tendency to move time to time, big time, like they start moving, progressing like 100 meters within a couple of days or weeks. And that brings huge amount of material to the lake. And we were interested if this surging is somehow influencing the community of chironomids, because it is doing it in regular times, so every, let's say, 20, 30 years, and we can always see the surging in the sediment. So this is the time when the surging appeared. This is a glacially warped sediment, so you will always have a winter lamina and a summer lamina. So it is a great sediment to study because it has this yearly resolution. And then we wanted to know if there is such thing like a glacier surge community, and how did the community or the surge influence the temperature reconstruction from this sediment sequence. So we took a surge layer, before surge and after surge layer, from I think 10 or 20 surge periods. And then we saw that actually the abundance and the taxonomic richness was very low during the surge. However, the community composition was not changing significantly, what was interesting. Then we did a temperature reconstruction on the surge periods are always marked with a arrow. And you can see that actually the surge temperature is about 2 or 3 temperatures lower in average than, that's the last slide, sorry. Average than the before or after surge, even though the climate was not changing. So the air temperature is not changing even. So meaning that other environmental changes can cause changes in your temperature signal if you are not carefully choosing your lake. So it is important to choose a right lake for temperature reconstruction or whatever reconstruction. The reconstructed factor should be the driving factor of your community and not other factors that are not related. Okay, so I have more, but I will not show you. I will just show you. Okay, I thank. Oh, yeah, see. So thank you very much. One more inspirational thing, that this is one of the oldest poems, and it was in the Japanese culture, and it was also inspired by a pond, which is a lake, a small lake, and I had to include this picture because we have so big chironomids in Slovakia, which is not true. It's a Photoshop, and it's not even a good Photoshop. I'm not good in it, but I thought that you should see how big chironomids we have in Slovakia. Thank you very much for your attention.
Unfortunately, we've run out of time for questions, but thank you so much, Ladislav. That was fantastic. Really interesting for me, not a world that I've heard much about. Thank you again. I'm sure you're sticking around for a bit, so if you've got any questions,
yeah, absolutely.
Please give them then. But other than that, see you next week or for the discussion next week.
Thank you for coming, and help yourself, please, while you can, with the chocolate, honey. Great. Thank you, Victor.